Now we can start. So, hi, good morning. Good morning, everybody. Welcome to the CPHMS webinar 7. So this time we have two excellent speakers, Dr. Jugnu Jain and Dr. Indumati M. So Dr. Jugnu is the CEO of Sapiens Bio and Saarum Sciences, and Dr. Indumati is a research scientist at LV Prasad Eye Institute at Hyderabad. So before we begin, I would like to call upon Dr. Surat, the Senior Research Associate at the Center for Predictive Human Model Systems, to speak for a couple of minutes about the center and the kind of work we carry out here. So, Dr. Surat, over to you.
Yeah, thanks, Sham. I'll just take like few minutes of your time and just give a brief this thing about what the center is. So thank you everyone for joining today on this Saturday morning. So 1 second. Okay, can you see the slides?
Yeah.
Yeah, we can see. Yeah.
Okay. Like Sham
Yes,
said, we are okay. So like Sham said, we are the Center for Predictive Human Model Systems at Atal Incubation Centre CCMB, and this center was established in 2019 as a collaboration between Atal Incubation Centre CCMB and Humane Society International India. And this was primarily established as a center to enable human relevant research in India. And towards this goal, we have been conducting several activities. So in the past 1.5 years, we have written 3 white papers on various human relevant research, including microphysiological systems, pathway-based research such as adverse outcome pathways, systems and computational biology. And these white papers have tried to understand what are the challenges in terms of funding research infrastructure to conduct this research in India. We also conduct several workshops and webinars, and this webinar is also part of the same initiative to increase more awareness in India amongst researchers and students for these methodologies, which are based on more human biology and not based on animal and not use animal models. So, and we also recently gave out a grant in the field of to build an adverse outcome pathway in the field of cancer last year, and 2 Indian scientists have been awarded that. And apart from this, we also regularly write reviews and popular science articles in journals and various news media outlets, again to increase awareness amongst both researchers and students and general community, scientific community about these methods. With this, I would just a brief intro. I would stop there and ask Sham to introduce our first speaker.
Yeah. Thank you, Dr. Surat. So now I would like to call upon Dr. Jugnu Jain. So Jugnu Jain is the CEO of Sapiens Bio and Saarum Sciences. So to give a brief intro about Dr. Jugnu, she has done a PhD from Cambridge University, and then postdoc from Dana-Farber Cancer Institute and Harvard Medical School. Then later on, she worked at various pharma institutes in the US, and then came back and set up Sapiens Biosciences in Hyderabad. So just to give a brief about Sapiens. So Sapiens is India's largest comprehensive biobank, which has a high quality, ethically consented and anonymized human samples. So with these human cell samples, it can be used for many diseases such as cancer, inflammation, immuno-oncology, cardiovascular disease, etc. So she has been an entrepreneur for a long time in this space of in life sciences. So we are glad to have Dr. Jugnu with us today. So Dr. Jugnu, over to you.
Yeah. Just a brief note, if you have any questions during Dr. Jugnu's talk, please type in the chat box and we'll take them towards the end of the call. And we'd request you not to switch on your mics. If there are any questions, please type in the chat box and
Can you see my screen?
Yes, we can see.
Yes, Doctor.
Wonderful. I really appreciate this initiative. I think one of the things I will add to what Shyam and Surat was saying is that one of the key roles I felt during our first roundtable that CPHMS did was also to bring together the community of people who are thinking about using translational models together. Certainly did not know of many of the people in India who are working towards this goal, and I want to grow this community together. So that is also a very, very noble and useful outcome of this. So I'm going to skip through one or two of the slides in the beginning because Shyam has already introduced us. But I think it's very important to know that we are building a very foundational system, a biobank, which in turn then enables a number of different applications. They could be diagnostics, they could be drugs, they could be better tools for many different research applications. So I'll give only a couple of examples given the limited time, but feel free to reach out on other things that might be of interest to you, which I don't manage to cover today. So we only use human patient samples or human normal samples for various testing of drugs, markers, targets, and so on. So it's two-dimensional, three-dimensional, and also intact tissue. So the essence of Sapiens, Sapiens stands for Homo sapiens, our human species, is to be a global biotech that employs human translational platforms for discovery and development of novel products for future patients. And towards that, we have built a systematic archive of ethically approved and procured samples that we then code to protect patient privacy and identity. And we use these cells and the stem cells that are in, for example, cancer tissues that we get to test various hypotheses. The differentiator is we are not a bone marrow or a stem cell regenerative medicine biobank. We are purely research and development biobank. So one of our key partners is Apollo Hospitals, which has 71 hospitals across the country as of now. And this partnership is where we get samples and data from Apollo, and we do have partnerships beyond Apollo now with PathCare DX and other hospitals. And then much of these kinds of applications are validated or invalidated using these samples and data. And the products of what we have done with our partners in turn goes back to hospitals like Apollo in the form of personalized diagnostics or treatment algorithms. So it's kind of a from the patient to the patient business model. As of now, we are already approved to collect samples from these locations within India. These are 10 locations, and you can see pretty much we cover most of India. So at the biobank, there is no geographical bias. There is no particular disease bias. I think the youngest patient from whom we have a sample is 3-day-old. And the oldest for whom I heard recently we have is a 101-year-old person. So it covers pretty much all diseases, all types of people who may come into a hospital. And we're constantly expanding the network. Hopefully by the time we speak next, we will also be in Guwahati. So we will be covering a little bit more of our Northeast. So the kinds of samples we get are, this is a snapshot of retrospective samples. This means these are leftover, literally medical waste blocks that are lying in various hospital pathology departments. The diagnosis is over, but they're required to keep these blocks for 5 years or 10 years, and after which they are typically discarded. So these are the blocks that we will lovingly curate and match with data. A lot of our internal work and examples that I will give will be in cancer. So this part is blown out for you to see the kinds of cancers that are prevalent in our country and hence in the biobank. So head and neck, breast cancer, generally women's cancers like reproductive organs for both male and female, digestive system, these comprise majority of our tumor blocks. We have all different kinds of samples. So they could be both tumor and normal. And in those samples could be blocks, could be fresh frozen tissue, could be blood, serum, plasma, could be DNA from any of these tissues. It could be synovial fluid, it could be CSF, many different kinds. So tissue is a very generic term that includes all bodily fluids and solid tissue as well. We do have access to a lot of urine and feces samples, which we have not yet started biobanking. Let's see how many requests we get. This is also an area from the point of view of microbiome, which we get requests for. So we're open for collaborations where perhaps we can write for a seed grant together to build capacity. In terms of cells, which is a lot of work we do, we make primary cells. So these are not cell lines, these are primary cells made from actual patient tumors that we get live. So a lot of our work is breast and glioma. I'll give you a couple of examples. And also blood cancers like AML, CLL, etc. We provide many different kinds of blood cells to companies and academia. These could be PBMCs, T cells, neutrophils, etc. And also from skin and other tissues, we provide keratinocytes and other cells like fibroblasts. So again, whatever you don't see here, but is of interest, and if it's not like a one-off, please contact us. We often do things, custom things for people's requirements. Very importantly, over the last 8 years since we've been around, we have been collecting a rich data set and increasingly partnering with knowledge partners that have bioinformatics capability, AI, ML. So we have treatment and outcomes data. This is in cancer. So for example, in breast cancer, we have data for 16,723 cases that are drawn from all those various sites that I have shown you in the previous map. This is longitudinal data, which is minimum 3 years and goes out to 5, and in a few cases up to 10 years. So it really helps for patient stratification and disease monitoring type of hypothesis validation. We work with companies that are looking for markers that can identify responders and non-responders to drugs. And I'll show you an example of that. And very importantly, this kind of data, we're talking to state government and organizations like RICH so that we can create data sets that can help drive public health policy for the state. So we, for example, can say, you know, your patients are coming from these districts, and in this district, this disease seems to be more prevalent, or, you know, patients are not able to afford this. You know, so the more the data that is able to generate some actionable knowledge, we would feel good about having created such a biobank. So what does data mean for different people? In our case, for cancer, this is the level of data we will have: patient demographics, their entire diagnostic history, their surgery details, their therapy. And it's not just chemo. So we'll have this detail for targeted therapies, radiotherapy, hormonal therapy, chemotherapy, etc. And then the outcomes. Where are they today and what is the disease state? If they recurred, what was the treatment for recurrence? I think somebody's computer needs to be muted. This is an example of a diagnostic, which is in partnership with a company in Boston, where they have a marker that can predict at the time of surgery in colorectal cancer, which patient will not respond to topoisomerase inhibitors, such as topotecan, irinotecan, etc. So our samples have been used by them to validate their marker. So on the left-hand side, you see 2 of our colorectal patient samples, colorectal cancer patient samples, where the patient is not going to respond and stains very heavily with this phosphoestin antibody. On the right-hand side are samples of patients that were given irinotecan and did respond. So because we already know which patient got which treatment, whether they responded or didn't respond, companies like Predictors come to us for getting these samples and data to see how accurate their predictions are. And this is something we've been doing in many cancer types overall. What would be of great satisfaction to me is if people in this audience already have prototype markers that are ready to be validated to know whether they will have clinical utility. Please talk to us. It'll be nice to see something useful come out of these research papers that get to a clinical trial. So this is a busy slide, but an important slide to show the various different ways we use the samples and data that we have gathered over the last 8 years. So on the left-hand side, the first vertical is OncoBlock, where this is the majority of our cases. It is FFPE blocks as well as fresh frozen material. And this is what is breast, glioma, colorectal cancer, etc. So this is used a lot by diagnostic companies and equipment companies for testing their antibodies and their research markers and testing whether, for example, recently I learned colorectal cancer has 20% patients who also express HER2. Now, HER2 was seen as a predominant marker for breast cancer. And yet there are medicines, very good medicines that can be used to treat patients in a very effective manner. So the more we know the molecular profile of the cancers, the better the treatment can be. So this is one of our main verticals. Next is live cancer cells. I'll show you examples of that, where we test molecules, could be small molecules, antibodies, nutraceuticals. We have herbal companies working with us. Then this area which has been growing, which is data alone for AI, ML, digital pathology applications. And then the right-hand side, which is non-cancer, where we do services for companies or academia for providing cells, human cells. Also, we do custom assays for proliferation, for cytokine production, many different kinds of assays. And then I'll show you an example where we use normal human skin, which is discarded from cosmetic surgeries, and how we use that to help many different kinds of companies assess the activity of their formulations and drugs on skin health and toxicity. So I'll give you a couple of slides on OncoPrime, which is using cancer cells from actual patient tumors. So what we do is we get this tumor tissue very closely working with pathology department. So from the operation theater, the sample will come, a large sample will come in normal saline, and a pathologist will gross that sample in our sterile hood and give us a small piece of the tumor tissue without compromising diagnosis. So that tissue is then dissociated and plated. And then from that, we derive two-dimensional monolayer cultures, as well as three-dimensional spheroid cultures that do not have any human serum and are not attached to plastic. So this is closer to how actual tumors will grow. These are multicellular spheroids. They go up to 150, 200 micron sizes. And in those then are these various functional readouts. So growth and death by various live dead cell assays, cancer stem cell quantification by flow, then by PCR looking at expression of stem cell markers, looking for surface expression of EMT markers, looking for migration and mobility, and then lastly, formation of soft agar, again, a three-dimensional assay in an anchorage independent manner, which is a hallmark of tumors. So this is a list. You don't need to read the list. Just this is to show you in the last 6 years or so when we started doing it, the list of about 20 brain cancers which we have cultured in our lab. These span the earliest grade benign desmoplastic ganglioglioma all the way to many GBMs. Again, many different age groups, male, female, etc. These have all been grown for testing drugs. Similarly, we've grown about 40 different breast cancers and another dozen or so AMLs and about half a dozen CLL, etc. These are some of the growth curves that show the power of 2D versus 3D cultures. Most of the life sciences community, academia uses cell lines, and most of the cell lines again are used in 2 dimensions. So they're grown flat with 10 to 20% FPS. We all know our human beings' cells don't have FPS and are not growing flat. Otherwise, we would not be standing up. So in 2D and 3D comparison, we can show different drugs have different effects. So here is a client compound, which is very active, whether you test it in the 2-dimensional or the 3-dimensional spheroid model. This is a very good anti-cancer compound. Here is a known chemotherapy, 5-fluorouracil. It's part of the standard breast cancer therapy. You can see it works quite well in 2-dimensional, shown here in this orangish color. So it inhibits proliferation going from 80% to 0. But it doesn't manage to block growth of 3-dimensional spheres because very likely it's not able to penetrate through multi-layers. And also it's been shown to not be effective on stem cells, which are responsible for regeneration of the tumor and metastasis. On the right-hand side is a known drug which somebody wishes to repurpose, so we had tested it for them, where this is a molecule which interestingly doesn't work on 2-dimensional cells at all, but works very well in 3-dimensional cultures. So this would be a molecule that is well suited for inhibiting stem cell regeneration and proliferation. So these kinds of assays provide a lot of insights into the mechanism of action of the drug and also how to test it in the clinic. Which drugs should it be combined? So for example, 5-FU that works well on 2D could be combined with this compound that works very well in 3D. So right up front, you kill the rapidly proliferating cells with 5-FU, plus a molecule that can kill stem cells to prevent recurrence and metastasis. So we provide a lot of very translational models. This is where we show how important it is to use primary cells and not be misled by only using cell lines. So here is a well-known cell line in gliomas, U87MG. If you look at the percentage of stem cells in this cell line, it's about 2% only. And even if you grow it in 3-dimensional spheres, that percentage of stem cell doesn't change very much. If you look at one of our primary cells, which is derived from that benign tumor, which is highly proliferative, right from the beginning, the percentage stem cells is 11%. So if you want to test drugs that have effect on stem cells, this will be a better system than this. And here it is responsive to growing as 3-dimensional spheres where you have cell-cell contact, and you can see almost a nice tight outer layer that is formed with these very healthy spheres. And you see the percentage of stem cell goes up 3-fold almost. So it's important that your system be robust if your readout is going to be meaningful. This is an experiment that told us early on that when we are growing cells from 2D versus 3D, you see upregulation of stem cell markers. So just ignore these two for a minute. If you look at the open versus closed bars, when you go from 2D to 3D cultures of the kind of primary cells we have, you see upregulation of several of the stem cell markers that are very well known to regulate growth. We work a lot with companies, as I said, and the data that we generate often forms part of their packages for regulatory submission. This is from one of our clients who's very happy with us, where we tested their drugs in gliomas, and the data that we generated was part of their regulatory submission package to FDA. I'll switch gears a little bit and talk about TrueSkin, which is another very different model system where we are not even culturing the cells. This is the intact skin. So there are a number of skin surgeries that happen that involve normal human beings. So there are abdominoplasties, There are breast reduction surgeries. There are skin grafts. There are amputations. There are many different kinds of these surgeries where people have skin coming out as a medical waste. So with the permission of the patient, we collect these samples. So we have multiple different uses of it. One is that we have small punches that we make in the skin, and I'll show you that in a minute. This is called an air-liquid interface or ALI model. So the skin is intact as it is. Then many companies require stratum corneum. So this is to look for permeation of drugs and formulations across the skin layer. So many people do 𝐒𝐞𝐬𝐬𝐢𝐨𝐧 assays. That is an artificial system that is created by putting cells and trying to create a layer. Here they will validate the results with using actual normal human skin. As I mentioned, we prepare various cells from it, and people buy these from us. And also we can provide skin-based DNA, RNA, protein, etc. for various assays. So the air liquid model is that we'll get a fat piece of skin. It'll look very healthy, normal like this. It will then be defatted, and small punches are made, 5 millimeter, 6 millimeter punches. And then they are placed in plates which have an insert on the top. So the insert on the top will have the punch standing up with the skin side facing up and the dermis side facing down. And the dermis part is in contact with liquid below. So it's an air. The top part is skin as it is exposed to air. The liquid, because the lower part of the skin is in contact with medium that will support its health and growth or health for the next 6 to 7 days. So that's called the air liquid model. You can see these small skin punches facing up. And then we apply various formulations or drugs or dermal applications as companies want. These are then kept alive for up to 7 days, and multiple applications can be made many times. And then there are multiple different readouts depending on the question the client wants to ask. It could be isolation of DNA, RNA. It could be looking at skin health and the gold standard assays, LDH. We do a lot of pathology, so H&E and other kinds of IHCs, immunofluorescence. We also do for skin permeability and absorption of drugs, and often we will then send the skin sample to the company to doing HPLC or LCMS to look at penetration of their drug. So this is something that really is as close to a human being as you would want. And instead of, poor hamsters or guinea pigs or something like that, this is actually human. So this is an LDH assay, which is the gold standard for looking for skin health. And you can say early on, there is hardly any LDH produced. The skin is very healthy. Over a period of time, as it's kept in vitro, the amount of LDH secreted in the supernatant goes up a little bit. So the skin is getting not so healthy after about 6-7 days, but it's very healthy for the first week. So it can be used for testing these kinds of applications. Here's a snapshot of some of the work we've done for, immunohistochemistry. So this is what an H&E image of the skin looks like. This is different antibodies, Claudin. This is immunofluorescence. So depending on the client's needs, they will give us their research antibodies, or we'll buy them and optimize them and then stain the samples for markers of their interest. So we do a lot of custom work. It's not off the shelf markers and things that are already done. These are again immunohistochemistry, where we had done optimization of 2 different antibodies, DCT and tyrosinase, and also provided them protein and other derivatives from the skin. How much time do I have?
Possibly 5 more minutes.
5 to 8 minutes.
Okay, so I'll just give
a few questions after the session. So yeah, because there are a few questions in the chat box, we also would like to take that. So
yeah, so then this will be the last or second last slide. We also do co-culture assays where we mix tumor cells of various kinds. It could be fibroblasts, tumor-associated fibroblasts, so they could be tumor cells. And you could mix it with blood cells from normal, healthy volunteers. So we do set up co-culture assays, which could be mixed lymphocyte reactions or immuno-oncology related assays. So I won't try to cover everything that we've done. Just stop at saying that the kinds of patient tissues and data that we have accumulated is extremely important in giving clues about why people have certain diseases, how they can be best cured, because no two human beings are the same. So this is an article that was written quite some time ago upon us. And I like their tagline saying that the cure is inside the patient. So it's the tissue that carries the clues about how to make diseases like cancer chronic rather than fatal. Thank you. I'll take questions.
Thanks, Dr. Jugnu, for the very interesting talk. And so there are a few questions in the chat box. I'll just read out to you so that everyone will know what the question is. The first question is by Ashwini, and he's asking, does the mechanical properties of stratum corneum changes after isolation? And if yes, can those changes show a different result compared to an intact skin for various compound testing?
Hi, Ashwini. It's possible there are some changes because once the skin has been cut, as you can imagine, it doesn't have the same tensile and other properties. But in terms of the permeability, I doubt that it will change that much because people are often looking to see multi-layer permeation. As I said, this is still closer to human being compared to animals, perhaps. So anything that you do here would still require the final validation in your test system. But I would guess it is closer to a human than other systems. But we've never tested that. So if there are mechanical engineers in this audience, which I'm sure there are, because this group attracts very diverse backgrounds, feel free to try.
That's true. That'll be a very interesting area of investigation to see how the mechanical properties. I think there are quite a few people who are testing that, but I'm not sure what the results are. So the next question is by Rohil. He has two questions. So first question is, what type of 3D cell culture approach is used for generating mammospheres? And second question is, what is the reproducibility of the results with 3D cell culture assays for drug sensitivity?
So the way we do it is we have very defined cell media, and we also use ultra low attachment plates, and we have no FPS. So the cells at early passages have a tendency to want to be together. So if you give them the right conditions, they will form these spheres. It's not as trivial. It did take us quite a few tries. And the defined media that we have come up with is also by publications, but also by trial and error. So there are quite a few tricks involved. Also, we have seen that the tissues that best form these spheres are from treatment naive patients. Obviously, if somebody has already undergone NACT, then the cells are not typically viable. They're very necrotic. So over a period of time, one develops a certain intuition for the cells. So our rate of success for growing these primary cells would be around 80-85% when it comes to breast cancer. The results are remarkably reproducible. Already in the biological systems, one can get 2 to 3-fold variation from experiment to experiment, but we always run standard controls, standard of care drugs as controls. So if you were to rank order your compound or your drug against the standard of care, that rank order holds from experiment to experiment. That is how one determines reproducibility. They are very robust.
Next thing is from Sandra from the European Commission JRC, and she has commented that she has the 3D spheroid culture, which from the brain donors, cancer cells. So she has also validated these cells for pyrogenicity testing. And she has also suggested that if you also get PBMC from the patients that you get your brain donor, you might also get additional ways of importance of early detection in cancers, for example, in solid tumors. And this might be an interesting to explore this avenue. So it's a comment.
I completely agree. We would love to work with people like that, because normally these protein or gene markers that you can detect in the blood are trying to find a needle in the haystack. So you need very sophisticated technologies to be able to detect early signals. And what we would like to do is, again, find out people who already have candidate markers. They could be any different technology. And since we have access to such kinds of matched tissue and blood samples, let's discuss how to validate or invalidate these early discoveries to have blood-based diagnostics, early detection. This is very valuable. We would love to partner.
The next question is from Manish, who was asking, how do you prepare cryobanks of 3D culture model? If so, he's asking, do you prepare cryobanks of 3D culture models? If so, how do you do so?
No, we have not at least done it so far. Whenever we try to store 3D spheroids, when we thaw them, again, you have to remove DMSO and many other things. So by the time they are thawed, the spheres fall apart. So if somebody knows how to do it, we'd like to know. But as of now, we store cells and create 3Ds from those cells after they have been thawed and plated as cells. Then in that defined medium under low attachment plates, the cells tend to come together as spheres. We don't freeze them as spheres. Whenever we tried to do it, as I said, they didn't stay as spheres when we thawed them.
Right. So in the interest of time, I'll just take one more question. So there is one question by Abhijit, who is asking, how do you see your 3D result as a good prediction in terms of treatment outcome for patients? Have you done some retrospective checks, and have you tried vascularization in the 3D models?
Yeah, so that's why I showed you 5FU. We've done this work with other drugs like cisplatin that are known to have differential effect. Some of the things that we would like to do, we haven't yet done, but quite a few of our results have panned out in showing that these drugs, this particular drug is not going to be able to block rapidly proliferating cells. We've also seen differential effects. Some drugs have worked very well in a couple of different types of tumors, but not in others. And that has also been observed from cell lines from other companies. So there is quite a lot of that relationship. We can discuss offline if you have a particular experiment in mind. What was the second part of it?
Have you tried vascularization in the 3D models?
Vascularization is very difficult. At this stage, we have not yet.
Thanks, Dr. Jugnu. So in the interest of time, I'll kind of move on, but there are a few, I think there are one or two questions still in the chat box, and maybe you can type in the chat box, Dr. Jugnu, while the next session is going on, just to save time. And Shyam, maybe we can move on to Dr. Indumati now, and if you could introduce her.
Yes. That is actually an insightful talk. Thanks, Dr. Jugnu. So now we move on to the second session, for which we have Dr. Indumati. So Dr. Indumati is a research scientist at L.V. Prasad Eye Institute at Hyderabad. She had done her doctoral training from the Center for Cellular and Molecular Biology, Hyderabad, also followed by her postdoctoral training at CCMB. And her expertise lies in the basic and translation research efforts towards addressing the problem of retinal and corneal diseases using different sources of adult and pluripotent stem cells. She was also in news for generating the first 3D corneal and retinal organoids in a dish. And apart from it, she was also awarded the B.M. Birla Science Prize for biology in 2016 for her contributions in the area of stem cell research. So having said this, we are glad to have Dr. Indumati with us. Over to you, Dr. Indumati. Dr. Indumati? Actually, you're muted. Yeah.
Sorry, can you hear me?
Yeah, we can hear you.
Thank you, Shyam, for the introduction. And first of all, let me thank Surat, Shyam, and Brinda for roping me in for this very useful initiative of spreading the awareness about the importance of patient-derived cells, not just in regenerative medicine, but also in understanding many disease biology and for drug screening. And I really enjoyed Jugnu's talk about she has really given a nice preview to how important the patient samples are. Though they were considered as a biological waste, at the clinical side, how a company or a basic researcher would find it very valuable. So with that, I will get into my talk. So I will mostly focus on applications of stem cells for regenerative medicine because our institute focuses on using patient-derived cells and stem cell-derived tissues for regenerative applications, with giving examples more in the eye field because that's the area that I work on. But nevertheless, the learning that we get can be extrapolated and applied for various other systems in the body. So with that, is my slide ongoing? Can you see my slides?
Yes, Dr. Indumati, yes.
So I couldn't advance it.
First slide only. So maybe you can stop sharing and start sharing again. I can only see the first slide on the list.
Yeah, I will do that. Yeah.
Yes.
So is it okay now?
Yeah.
Okay. So we are very familiar with 2D cultures,
Yes,
something
ma'am.
like that. So the 2D cultures, we do it every day, either with primary cultures taken from patients, like what Jugnu has been explaining, or it could be cell lines. They're nicely grown, as you can see, fibroblasts, epithelial cells, and neurons in the dish. But when you're talking about patient cells, you need a source, like from which part of the tissue that you can collect these cells, and can you expand them in culture? Because many of the somatic cells, unless it is a tumor cell, it's very difficult to expand in the lab. So here I have put in few examples of where the adult stem cells reside in the body. Like for example, you have a bone marrow, which is a complex tissue by itself. And you have limbus, which is a niche for the corneal epithelium. So your bone marrow is the niche for all your blood cells, and your mesenchymal cells that contribute to your bone, muscle, everything resides in the bone marrow. Similarly, your skin epithelial stem cells resides in the hair follicle. So you get a biopsy of the skin, you should be able to expand these cells in the culture. So as you can see here, the niche, it's not an individual component. It has a structural component. Each of them are unique with the crypt-like structures, and the stem cells reside deep inside. And the stem cells don't stay alone. There are different niche cells that are surrounding. So when you isolate these cells from the tissue and put them in isolation in culture condition, unless your culture conditions mimic the niche properties, it becomes difficult to expand these cells in vitro. So therefore, understanding of what niche does to the stem cells and replicating that in the dish is very important in establishing primary cultures, particularly patient-derived cells, if you want to use it for any downstream applications. So best example of applications of stem cells for regenerative medicine is the bone marrow, where people don't even expand them. So there is a step of purification and direct injection into the patients where it is required. So other tissue where an extensive in vitro expansion happens is for the skin grafting. So you can harvest skin tissues from unhealthy region and can apply it, expand in the lab to make a large sheets, and can treat injured areas. Say if it is a burn injury or an extensive skin abrasion caused by any accident or something. So these tissues can be regenerated and expanded, particularly because its adult skin cells has the ability to expand very well in vitro. So it's usually grown not just as monolayer of cells, but they are grown on suitable matrices such as collagen scaffolds to support these cells, and using suitable growth conditions, growth factors, these cells proliferate and make monolayers. And they can also be stratified because the skin is a stratified squamous epithelium. They get stratified and then transplanted onto the patients as patches. So these patches can be made, and then multiple patches can be used to treat it depending on the size of the wound. So you can see how the patches healed. So I would give you more examples on how such adult tissue-derived cells can be used for ocular regeneration. It focuses more on the cornea, which is the front part of the eye, and retina, which is the back side of the eye. There are many conditions wherein the surface of the eye gets damaged. Say there is an acid or alkali injury or burn injury that caused extensive damage to the surface, and then the cornea becomes opaque and the patient cannot see. So similarly, in the retina, there are also degenerative conditions which are inherited in the sense that the patients carry mutations in certain genes that are important for retinal function, and they degenerate and they, over time. So either an young child who was within 1 or 2 years after birth can completely lose vision, or it can be age related in the sense that a patient may have vision early in life but gradually lose vision as the cells keep dying. And there is no regeneration that happens in the retina, and therefore cell therapy or a gene therapy to replace defective genes are viable options. So to achieve this, we really need cells, some source from where these cells can be, donor cells can be harvested, and a mechanism to culture them to make transplantable tissues or sheets for therapy. So let's look at the corneal transplantations, stem cell therapy that has been going on at our institute for a long time, for more than a decade. So the cornea is the transparent part in the front part through which the light enters the eye, and it is transparent, but you see it as a black circle because of the iris behind is black, and the margin of the cornea between the white portion and the transparent portion is called the limbus. If you zoom in, you can see the nice crypt-like structures, and they are pigmented because they are melanocytes, which are niche cells that are present that keeps nurturing these stem cells. And you can harvest a small piece biopsy. Suppose a patient comes with a severe injury in one eye, the other eye can serve as donor tissue, and a small 2 mm biopsy is sufficient to expand enough cells to treat the affected eye. So here is the central part of the cornea, which is a uniform stratified epithelium, whereas the limbal region you see crypts and the stem cells reside deep in these crypts, and a small biopsy here will be the donor tissue. Here is a condition of a limbal stem cell deficiency. This is how a normal eye looks like, transparent. You can see the pupil clearly through the front part. In the limbal stem cell conditions, because the corneal epithelial cells don't exist and they are totally lost, the conjunctiva starts migrating in and the vasculature keep coming, and the whole cornea becomes opaque and light cannot enter and the patient cannot see. So this is a condition that can be easily treated. with limbal grafts from the fellow eye if fellow eye has some part of the limbus preserved. So what we do, we take a small biopsy from the normal eye, which is just 2 mm, 2 mm by 2 mm. And these biopsies can be cultured in the lab on suitable matrix. There are various matrix people normally use. And we use human amniotic membranes. Amniotic membrane is also a transparent tissue and an avascular tissue and has been used for a long time in the clinic for surface wound healing because it has hypoimmunogenic nature and doesn't trigger any immune reaction, and it's been used in the clinic for a long time. So we use amniotic membrane as the matrix. It has its own epithelium, which will be removed. We use enzymatic method to remove the epithelium. And then you make explant cultures on top of this denuded amniotic membrane matrix, wherein the explants will start, the stem cells in the explant will start proliferating, and you make monolayers of these cells. So this tissue, which is basically like a thin sheet of uniform monolayer of epithelium, can then be transplanted onto the patient eye. So this is how the well-established graft will look like. You have only 2 explants here. We normally put multiple explants in a particular donor tissue. So you see the 360-degree growth all around, and the growth from multiple explants will merge, and you get a nice monolayer. So this tissue can then go on to the patient. Here is the condition of a patient. You can see that both the eyes are affected. It's a case of burn injury, and most part of both the eyes are affected, but there are small regions here. You can see that is the limbal region is preserved. So a biopsy from that region could restore vision in both eyes. Here you can see that it's a complete opaque. So after 1 year of treatment with this particular tissue, what the clinician does is removes this damaged tissue and then places the limbal graft on top of it, either sutures it or puts a fibrin glue to fix the tissue on top and applies a bandage contact lens. Within a week, this tissue gets integrated onto the corneal surface and stays for a long time. And the vision is, if the underlying stromal tissue is normal, vision restoration is very efficient. You can see very complete transparency restored in this cornea. But if there are underlying damage in the stroma and there is scarring, then probably the patient may have to go for corneal grafting at a later point. So the overall success after treating about nearly 800 to 900 patients over a period of 10-12 years, we observe a success rate of about 71%, like in the sense out of 100 surgeries for LSCDs, about 71% of the patients. Somebody like to mute?
Yeah, you can go ahead.
So that's about adult stem cell applications, but there are conditions, as I said, for in case of retina, where the stem cells are totally absent in an adult retina. So all the development happens till the fetal stage, and beyond fetal stage, you really don't see any adult stem cells in an adult retina. So in there may be other tissues in the body where you really don't have any stem cells in an adult tissue, and you want an alternative approach. The best option is looking at induced pluripotent stem cells or the pluripotent stem cells, which can be either ES cells or iPSCs. So most of you might be aware that the ES cells are derived from the fertilized embryo, the inner cell mass. And these cells are pluripotent in the sense that it can differentiate into all 3 lineages. So basically any cell types of the body can be generated, provided we have a good protocol that will make make the differentiation program very efficient and successfully derive the cell or the tissue of interest. So iPSCs was another was a panacea for many of such applications, wherein so you can take adult stem cells or adult cells, so be it a blood cell or a skin cell, and it can be reprogrammed completely to an embryonic stage, and it doesn't have any ethical issues. So you can reprogram a fibroblast here, as shown here, by adding in some of the key transcription factors and reprogram them to an embryonic stage. Once it is reprogrammed to an embryonic stage, it is very similar to ES cells in terms of their potency, the sense that you can actually differentiate them to any lineage. So if I have a patient with retinal dystrophy and he has an issue with either age-related conditions or it's an inherited retinal dystrophy, a skin biopsy from the same patient can be reprogrammed to derive patient-specific iPSCs. In the sense that I have a model of retinal disease wherein there is a mutation in the line. And if I have to study the physiology of the disease, and this model will be of very useful. And at the same time, the genetic defect can also be corrected now with the advancement of gene editing methods in vitro. It is also possible to edit these mutations and see if the phenotype can be reversed. Or alternately, for an allergenic applications, a normal iPSC derived retinal cells can be given to the patients for therapeutic needs. So there is a lot of interest in using PSCs for deriving various tissues in the body. Say it's a mesoderm or ectoderm derived tissues like your brain or eye. So you can see the optic up here, or or kidney or intestinal organoids and pancreatic organoids. So there's a lot of literature in the past decade on both deriving tissue-specific cells from PSCs in monolayer cultures as well in three-dimensional organoid models. So what it involves, so you start with an embryonic state. So you need a method of pushing them to the cell type of interest. Here is so this is example is for the photoreceptors. So I comes from the ectoderm and the neuroectoderm. So you have to have a method of pushing these cells or differentiating them to each of these lineages. So this can be made possible by culturing these cells under unique differentiation conditions in the presence of different growth factors, which pushes or induces cells to get committed to each of these lineages. So this will be a stepwise differentiation, like in the sense you first push the cell ES cells to an ectoderm to an eye field commitment to a retina. So again, in the retina, there are different types of retinal cells that undergo degeneration. One is an RPE, which is a pigmented epithelium, which acts as inner cells for all the photoreceptors that actually are involved in light perception. So photoreceptors are neuronal cells, which are like extension of the brain, and they come from the neuroectoderm. And both photoreceptors and RPE cells come from a common precursor at the optic-vesical stage. So there has to be, if I'm interested in making photoreceptors, I need to have a different program or a differentiation method of enriching the cells or pushing the progenitor towards this lineage as opposed to this lineage. So these are very well worked out with the different stepwise protocols. A lot of publications has come out. Anybody interested can look for their own systems. But basically, overall, what it involves is taking in PSCs and then having a stepwise differentiation method to push them to each of the lineages and then to the tissue of interest from that lineage. And then once you derive that, you need to have an enrichment, because these are pluripotent stem cells. Differentiation is never 100%. You'll have contaminants of other lineages coming up, and you have to have a method of enriching them for cell type of interest. So I will give example of age-related macular degeneration. Macula is the central part of the retina which undergoes degeneration. Mostly this is an age-related conditions, and the RPE cells, I said the other type of retinal cells in the retina is the epithelium, pigmented epithelium. So that degenerates as the patient ages, and the degeneration in turn, because these are nerve cells helping in nourishing and clearing the supporting the retina also gradually degenerates and the patient lose vision as they age. So this is the central part of the retina. So this is what happens in these patients. The central vision, because the macula is the main central part of the retina, as the cells die, They will lose vision in the central part and periphery will be the normal. But as the disease progresses, even the peripheral vision will be lost and they tend to become totally blind. So this can be, as I said, it can be age related or it can be inherited. And ESCs, because retina doesn't have any stem cells, most groups around the world are looking at pluripotent stem cells, be either ESCs or iPSCs, for making retinal cells for therapy. So this is how the retina looks like. So you have a retina which is multi-layered, and the outermost layer is the photoreceptor layer, where the photoreceptor resides, and right adjacent to it is the RP cells, which is zoomed in here. These are cells that phagocytose all the shed photoreceptor segments and keep the subretinal space healthy and keep secreting factors and nourish the photoreceptors. So anything happens to the RP layer or to the photoreceptor cell physiology can lead to cell death and leading to disease phenotype. So in AMD, what happens, because the RP cells, this is the neuroretina, your RP cell layer is there here, and behind is the choroid, which has vasculature. Because the RP cells die, the choroidal vasculature starts coming in, and the vasculature sometimes tend to become leaky, and the blood leakage happens, and that can cause severe pain and vision loss in these patients. And there will be a lot of debris accumulation in the subretinal space, which can stress the photoreceptors, and the photoreceptors die. So what the strategy for cell therapy here is, the surgeon will clear the subretinal space, remove all and take care of the leaky vessels. And then the strategy is to transplant the RP cells either as a suspension or as a sheet of epithelium, just like how it is. It's a monolayer, it's not a stratified epithelium, it can be transplanted behind. So here is a schematic. As I said, it's a stepwise method. So pluripotent stem cells, which are typical ES-like colonies with distinct margins, you differentiate them in a stepwise manner at different stages. By about 2 months' time, you get pigmented epithelium coming up in patches. As I said, it's not an uniform culture. And from here, you have to enrich. You have an enrichment method wherein you can purify these specific cells, devoid of other undesired cells, to make monolayer of a highly enriched, more than 90-95% population of RP cells. So these cells can now go for treatment either as suspensions. So you can trypsinize and make a single cell suspension and inject them as small volumes in 10 microliter or 20 microliter into the subretinal space. In mice and rat experiments, people use very low volumes, or can go as a sheet, can be made into small sheet and delivered. So this is what I was telling you. So it can be single cell suspension, nicely pigmented cells, and or a sheet can go as a graft in the subretinal space behind, so that the RP cells that are lost here will get replaced, so that it will start supporting the retina and the retina will start functioning. So now that's about the applications of patient cells or stem cell derived tissues for clinical applications. So far we have been looking at monolayer cultures, and now in the past 4 or 5 years, we are increasingly getting publications and realizing the importance of 3D culture systems both in regenerative medicine and also in disease modeling and for screening drugs and etc. So as Jugnu was telling, these cells for 3D organoids can be derived from the patient samples from the biopsy, exactly like how Jugnu has nicely described, or it can be differentiated from ESCs or iPSCs. So the point that I want to emphasize here is, unlike a monolayer culture, which is just one single cell type, in an organoid, it's a complex tissue, wherein more than one cell types are present and they contribute to the organ or a particular structural arrangement of these cells, contributing to the tissue formation. So thereby they really mimic, or they're very close to the native tissue environment, wherein all the cell types that are there in the niche or in the surrounding environment are properly arranged in a fashion. This can be achieved either by manually, or it can be constructed in vitro, like by 3D printing, or by simple co-culture of 2 different cell types together, or in case of pluripotent stem cells, these cells can self-organize, because as I said, many of the cell types originate from common precursors. So when you provide a condition for differentiating them to a particular lineage, the common precursors can give rise to other related cells in a similar timeline, so that all the cells come together and self-organize to form a very complex tissue in the form of organoids. So here in this case, it is a self-organized organoids, whereas in case of either cell line cultures or primary cultures, we can actually do a co-cultures of 2 or 3 different cell types together, or grow it on a matrix, or 3D print the different cells in the desired pattern to create organoid models. So whatever it is, basically the organoid is a complex structure consisting of not just one cell type, but with multiple cell types in a proper organized in a proper manner, so that it mimics the native tissue. And it is a better model to study disease biology, because you have interactions of other cell types normally as to how it happens in vivo. So here I pulled out few images of how a brain organoid looks like. You can see nicely organized and laminated arrangement of these cells. These are self-organized stem cell derived organoids. Similarly, you can see the intestinal organoids. As I said, these are multiple cell types, the different cells that come together to form a complex structure. Similarly, liver organoids and lung organoids. And here is a complex organoid which is of brain and even the optic vesicles. You can see eye comes from the neuroectoderm. So they all come in a physically proximal locations, even in an organoid model. You can see the pigmented epithelium of an eye optic vesicle along with the brain organoid. And here is a retinal optic vesicle which is stained with some of the retinal markers. There are at least 6 different neurons in the retina. You can see that you can have markers for individual cell type, and you can see clearly that there are at least 3 different cell types nicely organized. This is not, nobody have told the cells to how to organize, because they know how to organize themselves, provided you give the right conditions in the dish. So this was nicely shown for the first time for the optic vesicles by Irako et al. And this is a Japanese group at the Riken Center in 2011. In suspension culture, basically you make an EVs and put them in right conditions. It's a suspension culture. Just like how the embryo develops with the 2 eye forming on either side of the organoids, you can see that eye primordia starts coming in. So this is the EGFP it's a reporter driven by eye-specific promoter. So you can see that the nice GFP containing precursors on either side, forming optic vesicles, which then imagine to form a cup. And the cup, you can see the nice neuroretinal marker being expressed. So now a lot of groups have published on this, and a lot of protocols are available. Similarly, not just for the eye, but for many other organs too. Our own lab also do work in this similar direction. So we push the cells to eye fields. These are just like you can see the popping eye. You can just by looking under the phase contrast microscope, you can identify these eye field clusters. And neuroretinal island forms a center, and the RP cells are naturally pigmented. Again, this can be easily seen by just looking at the microscope. And these cells can be easily isolated, sorted, make pure populations of sheets or suspensions. Organoids can be grown in suspension. Mostly the organoid, 3D organoid protocols involve suspension cultures, either in the presence of suitable ECM or gels, or complex matrices, or simple non-adherent suspension cultures, which allows the cells to organize, self-organize in a proper format. And they also laminate if it is a multicellular layers in the retina. They laminate, and different cells occupy different positions within the retina. So here is the cross-section of the retinal organoid. It's optic cup. You can see that the laminations, they're nicely. If you look at the fetal retina at about 18 weeks of differentiation development, it will exactly look like this. We also could show that not just the retinal primordia comes up, even the corneal primordia. In one of the previous slides, I was showing the brain organoid and the eye primordia coming next to each other. Here you can see within the eye structure, you can see the lens, you can see the retina, and also the cornea. Cornea is a transparent texture. You can see it as a transparent tissue coming up. So now you can actually dissect out each of these tissue types and go for unique cornea-specific culture condition or a retina-specific culture condition to further expand them in culture, or isolate cell types of your interest for whatever downstream applications you have. So what we have done is we have explored if these corneal organoids, in case of people with bilateral injuries where they need allografts because autografts are not possible because both eyes are damaged. So in that condition, can a stem cell derived corneal organoid be a source to make donor tissues? So we use the corneal organoids and dissect it and make nice sheets of epithelium on the amniotic membrane graft, just like how one normal adult limbal tissue graft will be prepared, and shown that these cells can nicely expand in culture, express all the markers that are unique, and now it's an unlimited source of tissue for regenerative applications, for allograft situations. And these tissues can be used for various things. If it is a patient-specific tissue, it can be used to understand disease biology, or it can be used for treating, checking for drug screening, and also for the treatment effects in vitro. So that's where the interest in 3D organoids, so not just in regenerative medicine, but also in using these small miniature structures for in vitro toxicology studies, drug discoveries, and for understanding disease biology, disease mechanisms, and also for using them as in vitro models to test infectious conditions. Even in a COVID situation, the lung organoids there are a couple of groups that are looking at using lung cells in vitro in 2D cultures or 3D organoids to investigate the viral infections. So here is an example of a polycystic kidney disease model. So it is a patient-specific iPSC line differentiated into kidney organoids. You can see how a normal kidney organoid look like. The size, it matters. So these are very miniature structures in the range of 1 to 3 mm maximum. So you can't really use them in direct application. You can't transplant. So if I say I can make corneal organoids, these corneal organoids cannot replace donor corneas because they are miniature in size. But they can use they have valuable applications in terms of being source cells, source tissues for isolating many of these tissue-specific cells. And we can make large numbers in one single differentiation. You can make about 30 to 50 organoids in a single dish. So that's the capability of stem cell-derived organoids. And the cystic, the organoids, kidney organoids, in case of polycystic kidney disease, you can see the cyst fluid accumulation and cyst formation, which proves the proof of concept. And now these models can be used for checking the efficacy of different drugs in vitro. Here is another example reported by Dr. Anand Swarup's group at NIH recently. It's a normal iPSCs, and here is a iPSC derived from a patient who carries a single point mutation in a gene called CRX, which is important for photoreceptor development. So all the photoreceptor precursors will express this protein, and you can see that in an organoid there is expression of this protein, but it's a non-functional protein in the mutant line. Level of expression is also low, but the organoids do develop in this model in the sense that the recoverin, which is a photoreceptor precursor marker, is equally expressed in both these models. That means there is no problem, even if this gene is mutated, there's no problem in retinal commitment or the tissue formation during development, but the maturation of tissue has a problem. Here they are showing that the opsins, which are expressed by the mature photoreceptors, be it cones or rods, both the markers they have checked, they are very highly expressed. These cells mature in vitro and they express these markers. whereas the CRX mutant model don't express any of this, suggesting that the commitment is not a problem when these tissues don't mature and these cells don't mature, precursors don't mature in this particular disease condition. It's proving the nice, providing a nice proof of concept. And interestingly, they have also used this model to test gene therapy. So they have a AAV vector which is expressing CRX and deliver this vector into this organoids after they have been derived. And they could show that these cells now, after CRX being delivered, they could differentiate and express the rhodopsin, which is mature cell markers. So thus they could the same experiments normally would be done in an animal model. Now the organoids could exactly replicate what expensive animal model study would convey. So basically the vectors they have derived are suitable. They're expressing in photoreceptors and they're able to restore a normal photoreceptor maturation in vitro. A nice proof of concept experimentation. Similarly, they have also, the same group have also reported that when you're trying gene therapies, you're delivering a gene of interest, but the gene of interest should be sufficiently expressed in the cell that you're interested. So people explore different promoters. Here is an example of trying CME promoter and a CRX promoter, which is again, CRX, I said, it's a photoreceptor specific gene and it's a precursor. So they have used this organoids to check the specificity of these promoters. So you can see the CME is an ubiquitous one. It's expressed in all cell types of the organoid, whereas CRX being photoreceptor specific, it's expressed only in the outer layer where the photoreceptor cells reside. Clearly confirming that the promoter is cell type specific and it can be used. Similarly, the AAV serotypes, which serotypes? Because there are serotypes that will, are more specific to one particular cell type. There are so similarly here they have tried AAV2 and AAV8, two different serotypes of the virus in an in vitro model and shown that AAV2 preference is efficient in infecting the retinal cells as opposed to AAV8. So therefore, most of the clinical trials now use AAV2 to deliver retinal genes and use many of the promoters like CRX or other retinal gene promoters for driving gene expression in gene therapy programs. So like so that's about stem cell derived organoids, which are self-organized ones. Similarly, self-organized organoids can also be made from cancer tissues, because cancer tissues, when you isolate cells from cancer, these cells inherently proliferate, and it is a mixed bundle of cells. You have vasculatures, you have different cell types in the tumor, and when you isolate them, you can isolate them as clusters and then grow them as organoids, or you can purify individual cell types and mix them together to form organoids. And these organoid models, cancer organoid models, can be used for drug efficacy studies, as Jugnu has already explained in detail, so I'll not spend too much time on this. And now we are coming up with the 3D printing and use of different scaffolding or the ECM materials and mixing of pure population, because with most of the primary cells, the availability of sufficient cells is a problem because many of the primary cultures don't expand very well. But in a stem cell based mechanism, you can really have a robust protocol for expanding large number of cells and purify them in sufficient quantities, so that you can really mix them together and use this kind of 3D printing method with suitable matrices to make desired tissues of interest and see the cells in a desired pattern that you wanted. So again, this is again about the 3D printing. You can see print out different types of tissues, be it is sheet or the complex tissue. And there is a workflow, as I said, that say this workflow shows about two different cell types and the ECM component coming together and being printed at different geometries to form a complex tissue as shown here. All this is possible now. So these will serve as wonderful in vitro models, replacing the need for animals. To expensive experiments can be avoided, and they can convey what exactly the animal models would convey us. And there are also tools and methods available now to even validate the functionality of these organoids. Say, for example, you have a kidney organoid or a liver organoid, and you want to test whether it is functional, whether it is metabolically active, you have your lab on a chip or an organ in a chip. So these are basically microfluidic based devices wherein you seed the cells or place the organoid, and then you add the metabolite in a different channel and see whether the enzymes or the cells can metabolize this product and give a readout. So this is now possible. And similarly, in case of neurons, you have multi-electrode arrays, whether earlier you have monolayer cultures of neurons plated on a dish containing electrodes. Now you can also have organoids, like say I have a retinal organoid. I can place it, I can shine light, keep the organoids in the dark and shine light and see whether the photoreceptors fire. And this kind of responses can now be recorded, and therefore it can also provide not just the source of cells, but also it can we can establish data that these organoids are actually functional and physiologically active to be and are valid models for testing various drugs. So with that, I will stop, and I thank all my group members, a lot of clinicians involved in the project, basic scientists, and my research fellows who are involved in the study. And funding from DBT-DST and the foundations, Hampley-Moore Foundation, Cognizant, and most of our animal work are happening at ICMR, Wizard and IN. And thank you very much. I'll be happy to take any questions from the audience.
Indumati, for the great talk. So if there are any questions, please type in the chat box. So there's one comment from Abhijit, although Dr. Jugnu has also answered it, but I'll still say it so that you can also give your opinion on it. So he is asking a general question that while we see fantastic work based on 3D spheroids, organoids, and organ-on-chip, why do you think that these models are yet not that popular among biologists and researchers? Is it because of difficulty of organoid culture compared to 2D cultures? And do you think a company or a facility supplying custom-made organoids might be useful to make the system more universally accepted?
Yeah, partly Abhijit himself has answered it. So it's the culture conditions or the in vitro conditions are really expensive for many people to replicate. And but even though I've said that, now robust protocols are now available for generating multiple organoids at this moment. And many companies are now interested in adopting this method and applying these organoids for various downstream applications. The advantage here is the large scale expansion ability and making a large amount of organoids that are required for a screening, for a pharmacological screening or something. It's like so basically it can reduce the cost of animal experiments, provided the organoids are sufficiently validated. That's another problem, because though you say that these cells, you can show by IHCs that different cells which are part of the organoids or normal tissues are present in the organoids, but that's not sufficient. So the organoids have to be functional. Unless the organoids are functional, they cannot be used for any physiologically relevant applications. So that is what is lacking in many model systems, many organoid model systems. Once that is developed, and if somebody has a robust method of showing the functionality of these ones, I think it will be widely accepted, and I think there will be an increased interest in this.
Kind of just adding on to that, Abhijit. So like you mentioned in the white papers that we wrote last year, so it did emerge like awareness was generally low amongst a lot of Indian researchers that such a technology exists. And this webinar series is also part of that same initiative that we are trying to do to create more awareness amongst both students and researchers, where we have Indian researchers come and talk about the work that they are doing in this field. And another aspect is not many of them are also, there aren't many training workshops in India, for example, to train more people in this field. So that is also something that we are initiating, and we'll are planning for a training, organoid training workshop which we had planned for this year, but then let's see, because of COVID and everything. It will be a hands-on session that we are planning. So yeah, and the other point about validation, which Dr. Indumati mentioned. So if I'm just checking the chat box if there are any other questions. Okay. So if
I had asked, maybe it was, we have access to a lot more skin than we end up using. So what is your requirement? Do you need more such samples, or are you self-sufficient?
Jugnu, we mainly focus on eye-related problems. So we deal with only ocular tissues. So we really don't work on skin's tissues. Basically, we don't have an in-house application for that, and we don't have any program running. So, but you could be talking to people who really need these models for. I think you're already doing it with pharma companies for skin-based products. So probably they would be interested to develop or use these models for their applications.
You were showing some skin monolayer kind of a thing for burn victims and so on. That's what made me think, maybe, is this somebody else's work?
No. It's just for the audience. So I just put it as an example from that's a well-known application of stem cell therapy. Other than the bone marrow transplantation, skin grafting has been widely used and for a long time now. So that's something people are totally aware of. That a stem cell or a cell-based therapy is possible for skin grafting. So it's just an example there. But we need to work on that. Yeah.
So I have seen at Tata Memorial, they take actually placenta. So the placenta is a non-immunogenic tissue for some reason, or it's very, very low immunogenicity.
Yes.
So they have these small little pieces that they autoclave and keep ready. So I just wanted to know if there are people who are doing something that is so useful and are short on material, if we can somehow help.
Yeah. So the tissue, the placental tissue that we use is the amniotic membrane. So the amniotic membrane is the chorion amnion. So from the placenta you separate them. So this part is totally avascular, and this has been used in both in skin grafting also, and also in ocular surface wound healing for a long time by clinicians. Clinicians are very familiar with this particular tissue usage, and it's been already acceptable. And so we use that. So people have shown that these cells are highly hypoimmunogenic and also has an immune suppression ability because of various components that these cells express, apart from being avascular. So that's the uniqueness of this tissue. And so when we started, the Japanese group first, when they reported the corneal epithelial graft preparation, they've exactly used the same model, and they've also used temperature sensitive matrix for that. So here, because the clinicians were already familiar with this tissue, it was started off using the amniotic membrane graft and worked perfectly well. And it was a success story.
You're right, it is amniotic membrane. I used the word placenta incorrectly. Yes.
So these membranes can be cryostored. So basically, not really cryostored in liquid nitrogen, but in -80 with the proper storage media. So they're aseptically processed. Either the eye bank does all the processing in our situation. So the eye bank collects the tissue from the hospital, and then process it with the same normal tissue processing SOPs that they follow, and make them into small usable grafts, like basically 3 to 4 4 centimeter squared grafts, and store them on in a cryo storage medium, so that you can use it for up to 6 months and in the vial. So it's always readily stocked. So clinician wants it with the limbal graft or without limbal graft, it's readily available with the eye bank, and they supply it. I think even Reliance is providing this for a lot of people, and I heard that they have a product of amniotic grafts also available for off-the-shelf ordering.
So just one more point which Dr. Abhijit has raised, if either Dr. Jugnu or Indumati can answer. So he's asking like what is accepted in terms of validation of these technologies. So either Jugnu or Dr. Indumati, you can answer this.
Go ahead, Indu.
So validation of organoid models, so say I can give you an example. So if you're looking at highly mature tissue function, which you cannot replicate in organoid model, say an organoid is at a fetal stage, you cannot replicate full maturity in vitro, then you can't use it as a model for predicting or understanding an adult tissue phenotype, right? So that is something that you have to have in mind, like what is the application that I have in mind that the cells can be of use. And secondly, the whatever application that you're looking at, you should have a measurable readout. If you're looking for a metabolic activity, and are you able to document functionality of these cells in terms of some assays that you can carry out, that it can convincingly say that the tissue and the cells that are part of the tissues are really functional and doing the job that you want to measure. So unless you have a readout or a method to assess that, I don't think the organoid even comes into play. Be it not just organoid, any cell type, any in vitro model, you should have an assay system and to show that the cells are viable and the right conditions. So the conditions may vary depending on the tissues or depending on the developmental problems that you want to address. So for an adult tissue-based assays, an organoid may not be a method. You may be well off if you choose an animal model to understand that. But for an early developmental defect or a cancer-based studies, an in vitro organoid model could really help.
So the way we do a lot of our validation is, again, to some extent by functional readouts, as Indu said. For example, the 3D models, they should be viable. So we do a very basic live dead. So the core of the 3D cultures should still be viable. We look for that layer that is a hallmark of a well-formed sphere. We look for ability of the epithelial and mesenchymal markers. Do they match from the original tissue to the 3D cultures that we have made? We look for upregulation of stem cell markers. The drugs that we use are the ones that are the second layer of validation because, for example, it is published that cisplatin works only on proliferating cells, that in fact use of cisplatin increases the presence of stem cells because it's able to kill proliferating cells but not stem cells. So using these drugs, we reproduce a number of results before we will take other people's drugs. Similarly, for example, in the mixed lymphocyte reactions, so we use dendritic cells from one patient and T cells or PBMCs from another healthy volunteer. When we set up the mixed lymphocyte reaction, we will have various stimuli. We will check with every control that you are really setting off an MLR because that stimulus is not able to stimulate only one component of the cells much. Similarly, we will use immunosuppressive drugs that are known to block certain component of the cells. And we will show that when you use cyclosporin, you block this arm. If you use dexamethasone, you block this arm. So we do a number of different tests to make sure that the system that we have set up is able to read out the requirement of that particular pathway or that set of cells for that drug that the client is interested in. So we may use different validation for different clients depending on their molecule of interest or their pathway of interest. The ultimate validation.
I also want
one more thing that is lacking so far, we have been hunting for people who would be willing to work with us to test our 3D models in animals. So the closest we have come to validation is in vitro, is the formation of spheroids on soft agar. That is the closest to in vitro tumor genicity that you can get to. But the other would be to have people who are setting up PDX models, where we could give our 3D cultures or tissues and cross-validate from the 3Ds that, are they able to form tumor in animals? So we are going backwards because this whole exercise is to try and get away from animals. But at the same time, there are certain tests one has to do to see whether you are really tumorigenic in an animal. So that would be something we would like to do if there is somebody who's willing to work with us.
I want to add one more point to the question by Abhijit about the validation thing of. So the usefulness of the organoid depends on how good the organoid is. So if there are interdependence of two different cell types, say an epithelium and a vasculature involvement, and if your organoid is not good enough and it doesn't have a vasculature, then probably it doesn't serve the purpose. So if there are two different cell types which are interacting and together contributing to a physiology, and both are present in an organoid, and you show functionality in terms of right assays, that the required physiology can be mimicked in your organoid model, then it serves the purpose. Because as I said, it's not a simple tissue, it's a complex tissue consisting of multiple cell types. If your organoid is not good enough to replicate the whole thing for disease modeling, then you should not go ahead. So that is where the field is moving on to improve the organoid model system so that it can be made closer to the native tissue, that it can be reliably used for all these assays. So that I think a lot of improvement will happen down the line, but at this moment, many of the organoid models are either one or two cell types coming together, but not really very complex. But I'm sure it is feasible with all the 3D printing and techniques coming into play with different matrices, and all that might definitely improve the organoid models and the reliability of the in vitro systems.
Right, yeah. Just to add on to that, I think that's where the field is going, like how we can make them more and more mimic the human organ. So there's a very hot area of research, like how we can, like someone raised the point of vascularization, of how do we include more immune cells into organoids, and also integrating multiple organs together to generate a human on a chip. And also, for example, including microbiota into these systems. So all these areas are areas of intense investigation, and there are a lot of labs globally that are working on this. And hopefully in the coming decade, we'll get there, where in more investment and research in these systems would lead to more generating systems that more and more mimic the human organ systems. So thanks a lot, Dr. Indumati and Dr. Jugnu for the great lectures. And if there are no more questions, I would just like to end on one small note. So there is another initiative that we are involved in. So in collaboration with the Joint Research Centre of the European Commission, Technical University of Denmark, and few other organizations, we are currently also conducting a survey to understand what are the issues, or what are the main issues that are currently perceived with the production and use of animal-derived materials and reagents. And this I'm just putting the link in the chat box, and anyone who is currently working in either human health, animal, or life science at any level, then if you could please fill this survey. So I'm just putting the link in the chat box. So it'll be great if you can provide your inputs on it. It's not a very long survey, and it's only going to take like a few minutes of your time. And because we're trying to understand currently a lot of the issues of reproducibility that are there in biological research, how much of that stems from animal-based reagents and materials that are used in lab experiments. And currently there are several initiatives that are working on non-animal based reagents and materials for various biological experiments. So how can we reduce the issues of reproducibility? So the survey would help us to understand those issues. So if you could just spare a few minutes of your time, and the link is there in the chat box. So with that, I would just thank you all, and Shyam.
Yeah, I thank you everyone for attending this interesting webinar. If I could request everyone, whoever can, open their video so that we can have a quick photo and then end this session. So if anyone, if they're able to open their videos, I would request to switch on their cameras so that can take a quick photo. Yeah, maybe a couple of more seconds. People are turning on their videos.
Is it done, Shyam?
Yeah, we're done. So thank you all. If anyone wants to reach out to us, I think you all have our email IDs, so you can reach out to us for any queries or any further collaborations. So having said that, we thank everyone who has attended this webinar and made this successful. So we'll also be sending the recorded links of the webinar soon, so that you can also share it with other people who might be interested in this particular topic. So having said that, thank you all, and hope to see you all in the next webinar. Thank you, everyone.
Thank you, everyone. Bye.